students, today we will discuss how do we use homologous recombination method for creating germ-like knockout mice or organism or somatic cell-specific knockout organisms. So we have P53 gene in mouse which suppresses tumor. It is a tumor suppressor gene. Now we want to remove this gene, P53 gene, so that we can use the transgenic organism, which is mice, for our research purpose. So in order to do that, first of all, what we do is we synthesize a vector. We incorporate 5' to 3' homology arm, and inside the homology arm we incorporate a neomycin resistant gene, and outside the homology arm we incorporate thymidine kinase gene. Thymidine kinase is sensitive to a drug which is known as gencyclovir. So this gene basically helps in the selection. We transfect the mouse. We isolate some embryonic stem cells from the mice, and then we transfect them with the vector that we have synthesized. So once they have been transfected with the homologous vector, what happens when there is there can be three possibilities when we transfect them. First possibility is that some of the embryonic stem cells will not get the vector. The vector will not get incorporated inside them. But another possibility is that the vector will go inside some of the embryonic stem cells. And the way vector gets incorporated, there are two ways for it. One is homologous recombination can occur. Another one is non-homologous recombination can occur. So let me tell you this, how homologous recombination looks like. If the vector gets incorporated into the embryonic stem cells, then and the recombination is homologous. That means between 5' to 3' of the mouse chromosome and 5' to 3', which we specially designed so that homologous recombination can occur of this vector. When homologous recombination will occur, then what will happen is the neomycin resistant gene will get exchanged with P53 gene. So ultimately what will happen is, this is vector and this is mouse chromosome. Now after combination, homologous recombination, the neomycin resistant gene will be exchanged. So it will come to the mouse chromosome and it is resistant. As already said, it is a neomycin resistant gene. And the P53 gene will go to the vector. Now the mouse chromosome does not have P53 gene, which is the purpose of our this recombination, because we want to create such transgenic mice in which there is no P53 chromosome, no P53 gene, and if there is no P53 gene, then we can use the mice for cancer research, because this kind of mice can easily get cancer. So we can check how a normal mice looks like and how the cancerous mice looks like. So after transfection, as already said, we will get some. So these black cells are indicating non-homologous recombinants, and the red are indicating transformed homologous recombinants. Both are transformed. So first of all, our first target will be to remove the non-transformed cells, because they are of no use for us. So for that we will do first selection, which will be positive selection. We will treat all the cells in a media which contains gentamicin or G418. Now the cells, those cells do not have neomycin resistant gene. untransformed because the vector has not entered them. That means they are lacking neomycin resistant gene. So those cells will die out in the presence of gentamicin G418. So that means they have been removed. That is the first selection which is possible. Now we are only left with two kind of cells: transformed non-homologous recombinants and transformed homologous recombinants. Now let me tell you this, how exactly a non-homologous recombinant cells will look like, their chromosome will look like the exchange will occur anywhere, not between 5' to 3' homology. It will occur anywhere. So both the genes will, if we will see here, in transformed recombinant cells, only neomycin resistant gene is incorporated. Thymidine kinase gene is still in the vector. So it has not been incorporated in the mice chromosome. But here in non-homologous recombinant cells, both the genes will get incorporated just anywhere, not specifically here. So that means non-homologous recombinant cells have both the type of genes, neomycin resistant gene and thymidine kinase gene. And that means these cells are sensitive. Thymidine kinase is sensitive to a drug known as gencyclovir. Thymidine kinase is used for the synthesis of thymine. If we treat these cells, non-homologous cells with gencyclovir, then it will inhibit thymidine kinase enzyme. And if this enzyme will be inhibited, then what will happen? The cell will be lacking thymidine. If there is no thymidine, then, because we know thymidine is a base, nitrogenous base. So if there is no thymidine, that means what? It means that the DNA synthesis will not occur. And if DNA synthesis will not occur, replication will not occur properly, then the cells will die out. So that's how we will select transformed recombinant cells. And we will remove transformed non-homologous recombinant cells. So the second selection will be negative selection, in which we will add G418 plus gencyclovir. As already said, due to the presence of gencyclovir, this gene will be inhibited. And once this gene is inhibited, all the cells will die out. So what we will be left with? We will be left with only transformed homologous recombinants. Then we will culture one. Now we have only these cells which are of our use. So we will culture them. And till morular stage, then we will implant it into the foster mother, where they can actually grow in the womb of the foster mother. Then later on, birth of P53 negative pups will occur. So these kind of pups we can use in cancer research to check differential expression of P53 protein. So friends, this is how we create knockout animals or organisms or mice using homologous recombination method.